Okay, here are the instructions for the microscope lab. This is task number 5. Watch this video, make sure you know how to use the microscope, and then do this lab. So the parts for the microscope are labeled on your handout. I just want to point them out to you on here. First one is the eyepiece. This has in it a 10x magnification. So even if you just have the eyepiece, then it's 10 times bigger than what you're looking at. The next thing are these 3 called the objective lenses, and these are what you will rotate to magnify what you're looking at. You always start from the shortest, which is the red, the one labeled red on these microscopes. The next is the stage. The stage is where you place your slide. The slide is the glass thing. And you can put your slide down on the stage and then put these metal clips on it to hold it in place. Below the stage is the light source. You want to make sure that the light is on, and you know it's on because you'll see the light and you'll know it's working if you put that line, press the on button and the line is down, the O is up. Make sure your microscope is plugged in, of course. There is this piece right here called the diaphragm, and you can rotate that. It regulates how much light is going through the slide. As you get better at this, you'll figure out when you need to add more light or not. The last 2 pieces that you need to know about are these adjustment knobs. And so you'll be using these adjustment knobs to move the slide closer or further away from the objective lens. You always start with the larger one, that's the coarse adjustments, and then once you have something that's sort of in focus, you switch over and use the fine tuning adjustment knobs. If you find something and then you call your friend over to come look and see what it is, they should always just use the fine one, the ones at the bottom, because if they use the coarse one, they're going to lose what you just looked at. Okay, so here I have my microscope. I am on the 4x objective lens, and I'm going to look through the eyepiece, and I'm going to use my coarse adjustment knob, and oh, look how you can see the beautiful tissue. Cool. All right, so now I've got something that's focused. Now I'm going to switch to my 10x objective lens. On our microscopes here, this is going to be the yellow one. Now, I might use the coarse adjustment knob, just a hair, or I might just use the fine adjustment knob so I don't move it too much. And yeah, there it goes, looks beautiful. Okay, and then the last piece is that you can go on to the 40x objective lens. This may or may not be focused for you. These ones are harder to focus. And so you may find that you have to go back to the 10x objective lens to actually see what you want to see. Once you have this, and now I've got a good look on this one. Once you have this, then you're going to go to your lab packet and draw what you see. You want to draw it exactly how you see it. Don't make things up just because you think it's supposed to look one way or another. And then label any cell parts that you can identify. The other thing that you must include is you must include the name of whatever slide you're looking at and the magnification. Right now I'm on the 40x objective lens. That's not 40x magnification. It's 40 times 10 for the eyepiece. So that is 400x magnification. If I want to go back and I want to draw it from the 10x objective lens, and that's perfectly fine, depending on what you're looking at, you want to call that now 100x magnification, because now it's only 10x here times 10x here. Okay, so now that you have all the instructions, make sure that you do this lab correctly, get 3 prepared slides, and then you can move on to the cheek cell.